Cancer requires durable, largely irreversible genomic alterations. Yet, experimental evidence shows these mutant genomes do not strictly dictate a permanent malignant phenotype. Cells accumulating chaotic structural flaws can still be normalized into highly organized tissue. When researchers map different highly mutated cancer cell lines on a scatter plot, we see them drifting far apart. Their transcriptomes are entirely heterogeneous. But when specific regulatory controls are applied, a distinct pattern emerges. The distinct clusters migrate along completely separate paths, yet they converge into a single dense cluster matching normal adjacent tissue. The physical genotype remains damaged, but the overarching regulatory state of the cell has been rewritten. The cells do not travel backward in time to recover a pre-malignant genome. The past is gone. Present-day regulatory conditions are altered so that a different, healthy state becomes accessible right now. Cellular restoration is a navigation at the present moment. Under the TCGS-SEQUENTION framework, this precise redirection of a pathological state into a tissue-compatible corridor is formalized as counterductive invariant recovery. A 2026 study by Gong and colleagues on colorectal cancer provides a precise biological anchor for how this framework operates in practice. Using an inference algorithm called Benign, the research team mapped single-cell transcriptomics from normal human colons. They filtered a massive cloud of data points into a predictive Boolean network map consisting of just 13 critical nodes. This network analysis identified an optimal control point. By simultaneously inhibiting 3 specific transcription factors, MyB, HDAC2, and FOXA2, the model predicted the malignant cells would stabilize into a normal enterocyte state. They tested this triple knockdown across 3 radically different colorectal cancer lines, HCT116, CACO2, and HT29. In all 3 cases, cellular proliferation dropped, normal lineage markers increased, and xenograft tumor mass shrank in vivo. The 3 target genes are deliberately altered. They are not biological invariants. They function merely as mutable molecular handles used to manipulate the broader network. The starting genomes of the 3 cell lines were completely heterogeneous, yet the intervention forced them toward an identical endpoint. The true biological invariant is the higher-order equivalence class the cells converged upon. Biological identity is sustained above the level of individual gene tokens. To understand the mechanics of this convergence, we have to look at how the Gong system mapped this cellular transformation as a topological network problem, evaluating the stability of the entire system at once. This graphic displays the model's unperturbed baseline landscape. Initially, a mere 12.5% of the Boolean state space leads to a normal enterocyte state. The vast majority of the map traps the cell in malignant attractors. When the triple inhibition constraint is applied to the model, the mathematical topography reacts immediately. The original 48 distinct valleys flatten out, merge, and erase the boundaries between them. Exactly 100% of the mathematical basin reallocates. It collapses into just 4 massive, highly stable valleys, all classified as normal enterocyte attractors. This topological shift canalized downstream effectors. It spontaneously opened over 2,400 distinct modeled routes, leading directly from a stem-like initial state to normal differentiation. The intervention did not force the cells down a single deterministic hallway. It reshaped the entire physical landscape so that normal differentiation became the only stable destination available to fall into. We see this exact mathematical restoration geometry in a parallel scientific domain, epigenetic rejuvenation. The mechanism is similar, but it aims at a completely different target. The foundation of this field relies on breaking cellular identity constraints using the Yamanaka factors, OCT4, SOX2, KLF4, and c-MYC. Total OSKM reprogramming carries immense danger. Pushing a cell too far erases its somatic identity entirely, resulting in a chaotic mass of uncontrolled pluripotency. To avoid this, researchers use partial, transient regimens. These shorter exposures successfully lower a cell's epigenetic age while strictly maintaining its necessary lineage identity. Cancer reversion uses differentiation controls to suppress a malignant state. Epigenetic rejuvenation uses transient plasticity factors to reduce a biological age metric. Because these two fields utilize opposite molecular handles to achieve distinct forms of identity-preserving restoration, they demonstrate that cellular age and cellular malignancy are entirely separate, non-equivalent coordinates. We plot cellular identity into a strict mathematical state space. The x-axis represents distance from lineage identity, the y-axis tracks malignancy, and the z-axis measures the cell's shadow age distance. The Gong cancer reversion trajectory begins high on the y-axis. The intervention minimizes the malignancy coordinate, driving the cell sharply downward into a stable target zone defined as the adult differentiated basin. A successful partial rejuvenation trajectory shifts laterally. It minimizes the shadow age coordinate on the z-axis, while carefully remaining aligned within a narrow vertical column representing its specific lineage. The primary hazard here is corridor overshoot. If rejuvenation factors are pushed too hard, the cell slides off the x-axis of its lineage entirely, falling into the hazard zone of dysplasia or teratoma formation. Both of these distinct trajectories must remain bound within the identity-preserving safety corridor. True biological restoration requires precise geometric navigation of these coordinates, not blind molecular forcing. This precise geometric navigation is a biological expression of the framework's overarching source architecture, the extrinsic constitutive law. This law is not an external ghost in the machine adding new information. Its expression is observed as selector naturality, the coherent convergence of highly diverse biological carriers into a single functional class. Because this relies on overarching geometry, the total number of mutations a cancer cell has accumulated doesn't automatically dictate its ability to be reverted. What dictates recovery is the integrity of the canalizing backbone, the underlying regulatory architecture that maps a genotype into a phenotype. If that regulatory backbone remains intact, a heavily mutated tumor retains the capacity to navigate back to normal differentiation. But if that structural corridor has been destroyed, a tumor with very few mutations is permanently cut off from recovery. Proving this geometry in a wet lab requires a strict experimental program. It starts with single-cell lineage tracing, definitively ruling out the chance that the treatment just killed the sick cells and left a healthy subpopulation behind. This chart plots a hysteresis curve, mapping control input against the biological state. Testing for this hysteresis is the ultimate requirement for proving counterductive invariant recovery, because it verifies the biological state is a true attractor and not merely clamped in place by drugs. Researchers must establish the forward threshold. As intervention dosage increases, the cell remains malignant until it hits a critical boundary. At that point, the malignant network breaks and the cell drops into the enterocyte basin. The critical test is the reverse threshold. The gene controls are totally withdrawn, reduced to zero. If the reversion is genuine, the cell remains flat on the bottom plateau, trapped in the healthy state on its own. This withdrawal stability must then be verified across the board. Transcriptome, protein, and functional assays all must agree that the cell is self-sustaining in its new class. Biological identity resides above the level of individual gene tokens. Cells are not historical tapes waiting to be rewound. They are spatial networks waiting to be intelligently rerouted.